As you can see, I finally got some grades posted. They're shown on the screen. And that's an interesting distribution. That's how things look right now. Also wanted to announce we have something I'd mentioned a week or two ago coming up that will be available to you for assistance with biochemistry. It's a new initiative in our department called the Major Groove. And the Major Groove is basically a set of evening help sessions for students who would like to come and get additional assistance in biochemistry in this biochemistry course. So Major Groove, they're still working out the details about the when and where, but the very first Major Groove will happen tonight, starting at about 5 o'clock. And though the location is not yet set, what I will do is I will send an email out announcing the location to everybody in the class as soon as that's set this afternoon sometime, and as well as the future times and dates as well. I think it's going to be pretty much Monday, Tuesday, Wednesday, Thursday. That's my guess. From typically about 5:30 until about 7:30, roughly thereabouts. So I will let you know. And I hope that you will use that resource. If it looks like you're using the resource, then of course we will continue to do it. And if it looks like you're not using it, that's fine too. But we probably will not continue to support it. So it'll be driven by your interest. Okay. Let's see. Go back to today's material, which is here. I'm going to finish up carbohydrates. And before I'll do that, I'll do the limerick of the day. Limerick of the day. To the courtroom the particle marched, with the public's outraged so enlarged. It's a sign of the times for atomical crimes that a neutron just cannot be charged. Oh, okay. All right. There's only a couple things I want to mention today. I'm not going to go through glycoproteins since I don't have figures, but I will just briefly mention a couple things about them. And then I'm going to say a little bit about signal sequence and transport, and then we'll move on to talking about signaling itself, which is a very fascinating topic. Last time I introduced the topic of glycosaminoglycans and proteoglycans and showed you some structures relative to that. That one I guess I won't show you. I will briefly mention to you. One of the other things that we have with respect to carbohydrates are what I call hybrids. And they're hybrids of carbohydrates with other things. Of course, one hybrid you've already seen are the glycosaminoglycans and the proteoglycans. Yet another hybrid involves making something I call an activated intermediate. Okay? We'll see other examples of activated intermediates, but an activated intermediate is a molecule that has a high energy bond, and it uses the energy of that bond in transferring a part of itself to something else. So it's a molecule that has a high energy bond, and it uses the energy of that bond to transfer a part of itself to something else. We'll see when we talk about the synthesis of glycogen that this molecule called UDP glucose is an intermediate in the synthesis of glycogen. And that's because UDP glucose has a high energy bond located within it. We'll see more about that a bit later. With respect to glycoproteins, there's another term I want to throw at you. Glycoproteins are molecules that are mostly protein, but they have short oligosaccharides attached to them. They differ from the glycosaminoglycans and the proteoglycans in that those were largely long polymers of modified saccharides. These guys are oligosaccharides that are attached to proteins. So they're fairly small, relatively small sets of saccharides attached to proteins. And these have a variety of functions. One of the most common of these functions is the blood types. The blood types differ from each other only in the configuration of the oligosaccharide attached to the blood protein. Okay, so that's a good example. And slight modifications of that oligosaccharide structure give rise to the different A, B, and O blood types that we are very familiar with. Oligosaccharides are typically linked through two different types of linkages to proteins. One's called N-linked, one is called O-linked. O-links are through serines, and N-links are through asparagines. Okay? Now, the last thing I want to talk about with respect to oligosaccharides are something called the signal sequence and transport. And these really relate to the movement of proteins through the cell. Okay? So proteins in cells, if you think about it, in a eukaryotic cell, we have different organelles. We have nucleus. We have the mitochondrion. We have other organelles, lysosome, for example, that all of which have enclosures within them. That is, that they are membrane-bound themselves. And how proteins get into them and how proteins move through a cell is a very important consideration. And one of those considerations for how they move is something called a signal sequence. That is, that there's what I like to think of as a license plate that's put onto a protein. That license plate determines ultimately where that protein is going to be targeted. There are two primary organelles of the cell that are involved in the modification of proteins to put those license plates onto those cells. They're known as the Golgi apparatus and the endoplasmic reticulum. And the Golgi apparatus is shown here, endoplasmic reticulum shown down here. And both of these are involved in making those N linkages and those O linkages that I talked about earlier. Okay? The O linkages are made exclusively in the Golgi apparatus. They're made exclusively in the Golgi apparatus, whereas the N linkages are made in both the endoplasmic reticulum and in the Golgi apparatus. So they're made in both places. And as we can see, the movement of the proteins through the cell occurs in a fashion that's rather like this. Here's the endoplasmic reticulum with pieces of it budding off. You can see these little round black structures on the surface of the endoplasmic reticulum. Those are actually ribosomes that are synthesizing proteins into the lumen, that is the central part of the endoplasmic reticulum. So as this part, within the endoplasmic reticulum, we start seeing some modifications. For example, we start seeing the N-linked modifications occurring. These parts of the endoplasmic reticulum will bud off and fuse with the Golgi apparatus and pass through a series of steps, ultimately targeted to their destination. One of those destinations can be being secreted outside of the cell. And so all of these kinds of modifications happen and are important. We see other modifications targeting them in the direction of the lysosome, lysosome being a degradatory organelle located inside of our cells. Okay. The modification of glycoproteins inside of the endoplasmic reticulum is interesting. It turns out that the oligosaccharide that is put onto the glycoproteins is synthesized apart from the protein that it gets attached to. So the oligosaccharide that ultimately becomes part of a glycoprotein is synthesized apart from the protein that it gets attached to. And it gets synthesized on the surface or on the end of this molecule right here called dolichol phosphate. So dolichol phosphate is a molecule that is very polar in nature, one end of it being very polar, the other end of it being very nonpolar. This is what we call amphiphilic, if you recall from our definitions. And this nonpolar part sits in the nonpolar portion of the cell's membrane, or in this case, the endoplasmic reticulum's membrane, very nicely, with the phosphate sticking outside of the endoplasmic reticulum. So if we think of the endoplasmic reticulum schematically, looking like this guy right here, okay, we can imagine then that in this little black region of the membrane of the endoplasmic reticulum, the dolichol phosphate is located, and the phosphate will be sitting on the outside surface of this membrane. There are enzymes that will start attaching onto that dolichol phosphate the sort of short saccharides that make up what looks sort of like a Christmas tree structure. It's a sort of a Y shape that it has. I don't have the figure to show you, but it's sort of a Y shape that this oligosaccharide has, and that's built onto that dolichol phosphate. After the thing has been built, it's very interesting what happens. What happens is dolichol phosphate, which has the phosphate and the sugar sticking out of the endoplasmic reticulum, undergoes a flipping process where the oligosaccharide flips to the inside. That is, the dolichol phosphate itself flips, carrying that oligosaccharide with it. And now the oligosaccharide is on the inside of the endoplasmic reticulum, and at that point it can be attached to the protein to make that glycoprotein that we're talking about. So that is the way in which, or one way in which glycoproteins are made inside of the endoplasmic reticulum. Okay. I mentioned blood antigens, and the blood antigens look like this. These are different saccharides that are attached there, and you can see that there's some very subtle differences in the difference between an O and an A, for example. Okay, so the only difference here being the attachment of one specific sugar here and a different sugar to make a B. And that's the only differences between the different blood types that we have. The reason that they're so reactive is these are recognized by different antibodies in our blood system. And if you have something that has, for example, if you have the A-type antigen, your body won't attack A, but if you put a B into that person, the immune system will recognize B as foreign and then attack it. And that's how the different blood antigens work. Okay. The very last thing I will mention is something, especially as flu season is here. I hope every one of you got your flu shot. If you did not, shame on you. You are health professionals. You should be getting immunized against these things. But flu shots are a good protection against the spread of the flu virus. Once you've gotten the flu virus, there are some drugs that will protect, I shouldn't say protect, but will help to reduce the impact of the flu virus on you, depending on the nature of the flu itself. And one of those is something, an enzyme that will inhibit an enzyme that the flu virus needs called neuraminidase. So the flu virus, in its life cycle, infects a cell, and when it infects a cell, it injects into a cell a series of short RNA molecules. Flu viruses are RNA viruses. And the reason we have different flu types and that they mix and match so readily is that there are so many different RNAs that are inserted into the cell during the infection. So you can imagine mixing and matching of these different RNAs can give rise to different types of flu. And in fact, that's exactly what happens. But anyway, the virus injects the RNA into the cell. It does this by virtue of the fact that it can attach to a cell surface. And the cell surface that it attaches to is a specific protein, a specific oligosaccharide that it recognizes on the surface. And this protein on the flu virus that recognizes it is called hemagglutinin. Okay, so you can see it right there, hemagglutinin right there. When the hemagglutinin attaches, that facilitates the entry of the RNA molecules into the cell. The RNA molecules in the cell get translated. They also get copied so that there are now zillions and zillions of little RNAs, and there are enzymes that will start to assemble a flu virus inside of the cell. It turns out that in order to get out of the cell, the flu virus requires action of this enzyme called neuraminidase. Neuraminidase is essential for the flu virus to exit a cell, and it's neuraminidase that is the target of an antiviral drug known as Tamiflu. So if you've heard of Tamiflu before, what it's doing is it's inhibiting the neuraminidase, and when the neuraminidase is inhibited, the flu virus cannot exit the cell readily, and consequently, the flu infection can be at least certainly slowed considerably. It can't be completely stopped, but it can be slowed considerably as a result of action of inhibiting the neuraminidase. Okay, so that's a lot of things about carbohydrates. I will stop at that point and ask if you have any questions. Yes, question. Yeah, can I clear up the role of hemagglutinin and neuraminidase a little bit more? Okay. So the two proteins that you see here on the cell, on the surface of the viral particle, are hemagglutinin and neuraminidase. The role of hemagglutinin is to find a specific oligosaccharide on the surface of a cell's membrane that it's going to infect. That hemagglutinin has a recognition and attachment point so that it can anchor the flu virus particle to that target cell. That anchoring is essential because that anchoring facilitates the entry of the RNAs from the virus itself. The role of the neuraminidase is that the neuraminidase is essential for the exit of the flu virus from the cell after the virus has been made. So we can imagine that these guys get injected. We start assembling flu viruses out inside of the cell. To get back out of the cell, that is to rupture the cell and get back out, the neuraminidase has to be active. What the neuraminidase does is it actually cleaves some oligosaccharides that are found in the cell membrane. That cleavage of these oligosaccharides is necessary for exit. If they can't be cleaved, the virus cannot exit the cell. And if the virus can't exit the cell, it cannot go and infect other cells. So by inhibiting the neuraminidase, one slows or theoretically stops the flu virus from being able to take over. It doesn't completely stop it, but it actually slows it down considerably. With any kind of infection that we have in our body, the main tools that we have are actually our immune system, because the longer we can wait, the longer we can give our immune system time to attack what's going on, the more likely we are to obviously recover from that. I talked about that with respect to Ebola, and that's certainly the case with the flu virus. So as we can slow down the flu virus, our immune system catches up and stops the infection from taking over our body. Okay? All right. Up top, yeah, question. Where does it get put in the cell? So this will actually inject right through here. It'll inject right through the cell membrane. Yep. The injection of nucleic acid into cells is a pretty cool phenomenon, actually. So, question, Justin.
What part of the flu virus actually injects the RNA?
You know, I don't know in the case of the flu virus. I don't know what part actually does the injection. Certainly for other viruses, they have specialized structures that will connect and actually make a little opening that opens that up and puts it in. I don't know how that is structured in the case of the flu virus. The classic virus for infecting cells that has been studied quite extensively in terms of the injection process is a bacteriophage known as T4. And T4 infects bacteria, and it makes this, actually has a stalk that is something that the viral DNA passes through during the infection process. Okay, well, let us move on to talk about signaling. Signaling is a phenomenon that is absolutely essential for a multicellular organism. It's absolutely essential for a multicellular organism. If we look at the phenomenon of signaling, the phenomenon of signaling is basically one cell of a body talking to another cell of a body. Some bacteria actually signal as well in a very primitive sense. That signaling that bacteria do, involves telling other bacteria, I'm here, I'm here. And the more signals that there are, I got the hiccups, the more signals that there are from one bacterium to another that says, I'm here, the more that the first bacterium knows that there are many others that are out there. It's called quorum sensing. So that in an infection, a bacterial infection, quorum sensing tells the other bacteria how many other bacteria are present. And that may be a factor in the life cycle of a bacterium. In the case of a multicellular organism, we can think of the specialized cells that make up us, our brain, our heart, our bones, our teeth, all these various things. And it would sure be nice for them to be able to signal and talk to each other. The nervous system that we have is one way of communicating information, But there are other ways of communicating information from one part of the body to another that don't involve the nervous system. And we're going to focus primarily on hormones. Okay? So hormones are going to be the primary things that we're interested in understanding in the phenomenon of signaling. Hormones are molecules that are released in one part of the body and travel to another part of the body to exert their effects. So they're released in one part of the body, travel to another part of the body to exert their effects. Okay? This schematically shows three different hormone systems that I will be talking about today and on Wednesday. And these, as you can see, the hormones in each case are listed at the top. So epinephrine is a hormone, insulin is a hormone, epidermal growth factor is a hormone. Epinephrine is a small molecule. It's related to the amino acid known as tyrosine. It's actually synthesized from tyrosine. Insulin and epidermal growth factor are both what we call protein hormones. That is that they are polypeptides that are synthesized by the body, and they exert some very big effects in terms of the target cells that they bind to. The first one I'll talk about is epinephrine, and you can see in the case of each hormone that's here that each hormone interacts with a specific receptor. The receptor for each hormone is located on the surface of the cell of a target cell. If a cell has a receptor for a specific hormone, then it can respond to that hormone. And if it doesn't have a receptor for that hormone, of course, it will not be able to take in that signal. Epinephrine is very important in, as it notes here, energy store mobilization. That's the adrenaline that I talked about the other day that allows the body to release tremendous amounts of glucose very, very quickly, as we shall see. Insulin has the effect of countering the effect of epinephrine. Instead of favoring the release of glucose, insulin favors the uptake of glucose by cells. And insulin plays a very important role in the body because, as I will tell you repeatedly through the rest of the term, glucose is a poison. The body treats it as a poison, and in fact, it acts as a poison. So the body has to manage levels of glucose very carefully. If it releases a lot of glucose, it better either burn it up or do something with it, because if it doesn't, there's going to be problems. And of course, we know some of those problems as they relate to the disease known as diabetes. Diabetes is a disease where people's bodies either don't produce insulin or don't respond appropriately to insulin. And as a consequence, their blood glucose levels go very high, very low, very high. They're very sporadic. And that very high can cause some enormous problems because, again, glucose is a poison. Epidermal growth factor, the last of the hormones there, is important in the decision, helping the cell to make the decision, do I divide or not divide? And we'll talk about that in just a bit. Okay? In the case of epidermal growth factor, we see that it manifests its action through the expression of growth-promoting genes. Okay? These first two guys are sort of immediate acting, release glucose, take up glucose. Whereas epidermal growth factor is a longer-term phenomenon, and it's involved in the control of the synthesis of specific classes of genes. And we'll see more about that. Okay. I've mentioned before that, at least briefly, that cells are what I call control freaks. They have to have the ability to, if they have the ability to turn something on, they also have to have the ability to turn it off. We can see here that this schematic diagram, starting at the top and moving to the bottom, is the way in which hormones exert their effects. The signal is a hormone that's released in a part of the body, and it travels through the bloodstream to target cells where the reception occurs, and that's where a specific receptor binds to that hormone. The receptor then causes some changes to happen inside of the cell as a result of binding the hormone. And those changes are what we call transduction. And transduction literally means moving information across the cell membrane. We'll see how that occurs in just a few minutes. That transduction results in responses, as we shall see. Those responses can be enzymatic. as in the case of the epinephrine, those responses can be regulating gene expression, as we see in the case of the epidermal growth factor. Now, as I said, it's important if we turn these systems on that we also have ways of turning them off. And the blue arrows that are showing here are all giving feedback to the body saying, okay, we got your information, quit sending us more hormones, quit binding more hormone receptors, quit introducing this signal into cells. So the blue arrows are countering the effect of the signal that's coming down, ultimately moving downwards. Okay. Hormones in the signaling system in the body are something that we call first messengers. They are the first thing that the body puts out to signal something needs to happen. They're the first messenger. Okay? Well, that implies that there are second messengers, and the second messengers, in fact, are molecules that are made inside of cells as a result of a hormone binding to a cell's receptor. So a second messenger is a molecule that's made inside of a cell in response to a binding of a hormone by a cell's receptor. There are numerous second messengers, and you can see they take quite a wide variety of chemical forms. The first one that we'll talk about is cyclic AMP. We've already mentioned it briefly. There's also a related compound to cyclic AMP called cyclic GMP, and we will mention that one actually on the very last day of class. We'll see how that plays out. Calcium can act as a second messenger. Inositol 1,4,5 triphosphate, or what you're more likely to call it, IP3, is the second messenger, and diacylglycerol is the second messenger. And we'll talk about all these over the next couple of days. Okay. Well, I want to start by talking about a class of receptors that are found on the surface of cells. And this class of receptors includes the receptor that binds to epinephrine. Okay? These receptors all have a similar general structure, not a similar specific structure, but a similar general structure. And they're called 7-TMs, and I'll show you why they're called that in just a second. But 7-TM receptors control a wide variety of things inside of cells. And again, this is not a list for you to memorize, but you can just see some of the many things that 7-TM receptors help to control. Everything from the control of blood pressure, embryogenesis, smell, neurotransmission, a wide variety of things that 7-TMs can do. 7-TMs get their name by virtue of the fact that, first of all, they are membrane proteins. If you're going to bind something outside the cell, you had better be located close to the outside of the cell, and the membrane, of course, would be the place to be located. A 7-TM gets its name from the fact that the protein has what we call 7 transmembrane domains. That's the TM part. So a 7-TM crosses the membrane 7 times. So we see here's the N-terminus of the protein. There's one domain crossing the membrane. There's a second, third, fourth, fifth, sixth, seventh. So 7-TMs will all have 7 transmembrane domains. And obviously similar structures and all, of course, located inside of membranes. All receptors for hormones are located inside of membranes. All hormone receptors are located in membranes. The last figure was a schematic representation of a 7-TM. In reality, 7-TMs look more like what you see here. They still do, in fact, cross the membrane 7 times, but they're not organized in a nice linear fashion like you saw in the last figure, but instead commonly have a structure that's sort of barrel-like, as you can see here. There's the outside of the membrane shown on the top, and there's the inside shown on the bottom. The inside, of course, is where the cytoplasm is located, and the outside is where the hormone is going to bind. So the binding of the hormone on the outside of the cell, that is up here, causes a slight structural change. There we go again, guys. A slight structural change inside of the 7-TM, and that slight structural change results in some drastic changes on the inside of the cell. The molecule epinephrine, as you see here, is the one I'm going to talk about first. As I said, it is related to tyrosine. Tyrosine, of course, is an amino acid that has a hydroxyl group sticking off of a benzene ring. You can see that this guy has been modified a bit to give it the structure that it does. Epinephrine, as I noted, is also called adrenaline, and I use the terms interchangeably. The action of hormones to cause changes inside of cells is very interesting. Okay? It goes through a series of steps, and the series of steps that we will talk about are shown right here on the screen. I'm going to step you through it kind of quickly. Okay? What we see is the process that starts at the very top with the binding of epinephrine by a cell receptor. Okay? Well, when is epinephrine released? Epinephrine is released when the adrenal glands get a signal from the brain, we're in danger. We got to get out of here. We're in danger. Okay? So epinephrine is released. That causes ultimately, as I mentioned last time, that ultimately causes release of a giant amount of glucose, and we'll see later how that process occurs. We're seeing the first half of the process here. The last half of the process we'll talk about when we talk about glycogen metabolism. Okay? Well, the first half of the process starts with the release of epinephrine into the bloodstream. There's your first messenger, epinephrine. The receptor that epinephrine binds to is called the beta-adrenergic receptor. That's the name of the receptor that epinephrine is binding to, the beta-adrenergic receptor. Binding of the epinephrine by the beta-adrenergic receptor causes a change in the structure of the 7-TM. This is a 7-TM, of course. And that 7-TM is interacting with something called a G protein. A G protein, the letter G. Okay? A G protein gets its name from the fact that it binds to guanine nucleotides. So normally in the cell, the beta-adrenergic receptor is sitting there, snuggled up closely with a G protein. The G protein has 3 different subunits called alpha, beta, and gamma. So they're sitting there minding their own business, and epinephrine comes along, and when epinephrine binds, everything starts to happen. The binding of epinephrine by the beta-adrenergic receptor causes the G protein to do 2 things. First, it sheds the beta and gamma subunits. They're gone. They're out of the way. The beta and the gamma subunits are covering up a binding site on the alpha subunit that would allow it to interact with this enzyme, adenylate cyclase. When the beta and the gamma go away, the alpha is free to interact with the adenylate cyclase. So the first step in that process is getting rid of the beta and the gamma units. The second thing that happens upon binding of epinephrine by the 7-TM is that the GDP that you see in here is kicked out and replaced by GTP. Notice that it's kicked out and replaced. There's not an addition of a phosphate to make it a triphosphate. There's a replacement. GDP is replaced by GTP. Now, what GTP does is it gives this alpha subunit what we call activity. It acts in an active fashion. The combination of the fact that the beta and gamma subunits are gone and this guy is loaded with GTP allows the alpha subunit to interact closely with adenylate cyclase. Adenylate cyclase is an enzyme, see it's an ASE, that's found in the membrane of cells. And the binding of the alpha subunit with GTP causes adenylate cyclase to become active. The catalytic action of adenylate cyclase is to make cyclic AMP. So we saw cyclic AMP the other day. Now you're seeing how cyclic AMP is made. It's made when adenylate cyclase is activated, and adenylate cyclase is activated by a G protein. Well, you've already seen what happens when the cell has adenylate cyclase, because adenylate cyclase can bind to the protein known as protein kinase A, and that causes it to go from an inactive state to an active state. We have started a signaling process. The signal started out here with epinephrine. It continued through the receptor, through the G protein, through the adenylate cyclase, through the cyclic AMP, and it's going to continue through the protein kinase A. Yes, question? It interacts with the G protein. Is that what you're talking about here? So, her question is, when the binding of epinephrine causes some changes, does that affect, I think the question is, does it affect the interaction here? Normally what's happening in the cell when there's no epinephrine bound, all right, this 7-TM is sitting here snuggled up close with this G protein just like you see. The binding of epinephrine causes these two changes to happen: the release of the beta-gamma subunits and the loading of GTP in place of GDP. Then, now this guy can interact over here. So that's a necessary step. If this epinephrine doesn't bind, then this rest of this process can't occur. Okay? Yes? Okay, so the question is, where is the GTP being loaded? It's being loaded right in the middle of the alpha subunit. Right in the middle of the alpha subunit. Up top. Question up top. Yep. The G protein is not located in the membrane. As you can see, it's peripheral to the membrane, and it's interacting with the beta-adrenergic receptor. It's free to slide around. That's correct. Yes? So our 7-TMs located in an organelle. There may be some 7-TMs that are actually in organelles, but they would not function in signal transduction. So that common structure we see in a variety of functions that are present inside of cells, but not in the signaling process. Say it again. So the 7-TMs are primarily located in the plasma membrane of the cell, as you see here. Yeah. Question over here. Why did they replace, good question, why did they replace GDP with GTP? Okay, so she's getting ready she's anticipating the next thing I'm going to tell you. First of all, I did say that the adding of GTP causes this to be active, and the activation of this is necessary for the activation of this enzyme. I can't tell you why they replaced it instead of just adding a phosphate to it, but I can tell you that they do that. There actually is a bit of an answer to that, but suffice it to say, again, the mechanism, we weren't there when this evolved, so we can't really say, right? But there actually probably is a reason why it doesn't just simply add phosphate, okay? It turns out that G proteins are interesting proteins. It turns out that G proteins are really bad enzymes. They're really bad enzymes. Okay? And the fact that they're really bad enzymes is really good. Why is that really good? Okay? Well, let's understand what G proteins do. They do catalyze reactions. And the reaction that they catalyze is the breakdown of GTP to make GDP. That's kind of dumb. Well, actually, it's not. If they were really good enzymes, what would happen once you loaded a GTP on here? It would immediately break it down, and the signal would never make it over to adenylate cyclase. It turns out that because they're bad enzymes, it takes a while for that breakdown to occur. And because it takes a while, this guy is free to float over and activate that before it breaks down the GTP. Why does it have the ability to break down GTP at all? The answer to that is that's one way of turning off the signal. We're going to see that the synthesis of cyclic AMP is a very, very powerful thing for cells. It's a very powerful thing for cells. When cells make cyclic AMP, giant changes in the cell happen because the cell's going, whoa, they're wanting me to do something with glucose. And if I'm a muscle cell, I am breaking down my glycogen. And if I'm a liver cell, I'm breaking down my glycogen and I'm making glucose. Cyclic AMP has drastic effects. So the cell has to be very careful to be sure to turn off signals that it turns on, not let them stay on too long. Imagine that one of these lights that we had up here, okay, had an enormous amount of electricity that it took, such that by turning it on, the power bill for this room went up enormously. Maybe it would heat this room a little bit. That might be nice, huh? So that the power in this room went up enormously. We wouldn't want to leave that light on for too long, right? Same thing with the G protein. We don't want that G protein to be activated for too long. So what it does is it actually turns itself off. Because when it cleaves GTP, it makes GDP. And in GDP, it picks up the beta and the gamma and goes right back here and waits for another signal. It's a self-turning off mechanism. Now, that's one cool thing. I'll tell you another cool thing. I told you cyclic AMP was very powerful. We've already made cyclic AMP by this point. Now we've got this molecule here that is telling the cell, get some glucose, get some glucose, get some glucose, and put it out in the bloodstream so the muscles have energy to get us out of this danger that we're in. If we don't have a way of getting rid of that cyclic AMP, we still have a problem, right? This is a cool thing. The cell has a very neat way of getting rid of cyclic AMP. How does it get rid of cyclic AMP? Well, it has an enzyme floating around inside here all the time called phosphodiesterase. Phosphodiesterase. I'll put it in the notes so that I won't spell it here. But phosphodiesterase breaks down cyclic AMP. That's good because when it breaks down cyclic AMP, now protein kinase A goes back to being the inactive form. And the cell, which has been going, make glucose, make glucose, make glucose, no longer has a signal telling it to do that, so it stops making glucose. Now your blood glucose levels fall back down to a normal level. That's good because glucose is a poison. Okay? Everything is fine. Phosphodiesterase is a very important enzyme in this scheme. Phosphodiesterase, it turns out, is inhibited by something that you guys drink every day. Coffee. It's inhibited by caffeine. So when you inhibit phosphodiesterase, what's happening to you? You're inhibiting phosphodiesterase, your cellular cyclic AMP levels are increasing, your blood glucose levels are increasing, and you are getting a buzz. That's how you get a buzz. It's one way you get a buzz. There's many ways of getting a buzz. Okay? Does that make sense? Yes, question over here.
So, when epinephrine binds, the G protein kicks out GDP
Yep.
for GTP, but eventually GT.
Yeah, so our question is, when epinephrine binds, GTP gets loaded, and then eventually this guy breaks back down to GDP and comes right back over here. Yep. So it's a self-turning off mechanism. We'll see this self-turning off mechanism also happens with other G proteins. In fact, that's a characteristic of G proteins. And if a G protein is involved in telling a cell to divide, and something screws up and it can't turn off that signal, what's going to happen? Cancer. Okay? So this mechanism of turning itself off is important. If you screw up the mechanism of turning it off, you could imagine a cell is going to have some serious problems. In the case of this cell, it would probably just burn itself out. It would burn up all the glucose it had, and then it would be no more if it didn't turn itself off. But if this G protein were controlling a decision to divide or not, that might be a very big consideration for a cancer. Other questions? Yeah, question there. Yep. After epinephrine binds, is it released? The answer is yes, it is released. And it's also degraded. Okay? And we'll see, I'm not going to have time to do it today, but we will see next time a little bit about how there's one more control built into this, and that is to help the receptor to turn itself, to be turned off. We don't want the receptor to be stuck in the on mechanism. And no, I'm not done yet, folks. Yes?
In all of the pictures of the 7TMs, the amino end is outside?
Yes?
Is that always the case?
On 7TMs, is the amino end always outside? As far as I know, but that isn't necessarily a rule. The rule is that there's 7 crosses. Yes?
So how does caffeine inhibit phosphodiesterase? Allosteric, suicide?
How does caffeine inhibit phosphodiesterase? It's a good question. I believe it's allosteric, but I don't know. It's definitely not suicide, no. Yeah, up there, question. Are there any harmful long-term effects of drinking coffee? You know, people have asked that question a lot, and the more that they've studied caffeine, what they have found is that for the most part, caffeine is actually pretty good for you. It actually appears to have a protective effect in liver. And while I wouldn't recommend going out and drinking 30 cups a day, even moderate to semi-high levels don't seem to have any major negative effects. So that's an interesting observation about caffeine. Yeah. You can feel good about drinking your coffee. What I wouldn't do is go drink coffee. Let's think about this for a second, folks. People don't drink coffee. They drink god-awful, sugar-loaded, cream-loaded, right? Think about this. The buzz that you're getting is coming in 2 ways. One, your body is dumping glucose, and what are you dumping into your body? Glucose. Oh my God, you don't want to do that. Glucose is a poison. Let's sing a song and get the hell out of here. The song today relates to something I talked about last time. It's called hyaluronic acid. Hyaluronic acid, acting almost magically, placed just beneath the kneecap, lubricating the debris. Better than joint replacement, simple as 1, 2, 3. If it can stop the aching, you will get to keep your knee. When the pain is getting bad, try not to be sad. Just go out and have a talk with your orthopedic doc. Beg him to use the needle. To not do so would be a crime. Hyaluronic acid, working where the sun don't shine. See you guys on Wednesday.